Hello friends, in this module we will talk about lysogeny with respect to lambda phage, which is a temperate phage.
Bacteriophages reproduce by the lytic cycle, which leads to the destruction of the host cell. Some bacteriophages, like the lambda phage of E. coli, follows a totally different life cycle. In this cycle, the viral genome is injected into the host cell. But instead of arresting the host cell, the viral genome is integrated into the host cell genome. Every time the host cell genome replicates, the viral DNA is also replicated along with it. Thus, the viral DNA gets a free ride along with the host cell. The viral DNA can remain in the host cell for very long periods. This relationship between the host cell and the phage is called lysogeny. Such host cell bacteria are called lysogens, and the phages which can enter into lytic cycle or lysogeny are called temperate phages. The phage genome, when integrated in the host genome, is called a prophage. Lambda phage is a common phage which can get into lysogeny with its host cell, the K12 strain of E. coli. Hence, we will study lysogeny with reference to lambda phage. The lambda phage has an icosahedral head, a tail, and a thin tail fiber which is mainly responsible for recognition of the host cell. The lambda phage genome is a linear double-stranded DNA molecule 48.5 kb in size with single-stranded regions at the 5' ends. These single-stranded tails are 12 nucleotides long and are complementary to each other. These single-stranded tails are also called sticky ends. Hence, they can base pair with each other and undergo circularization. Lambda phage can lead two different life cycles. It can lead the T4 bacteriophage-like lytic cycle, wherein upon injection of the DNA into the host cell, the DNA undergoes circularization. The phage genes are expressed, phage DNA is replicated, and many phage particles are produced and are released by lysis. On the other hand, it can follow lysogeny, wherein its DNA remains integrated in the host genome without any harm for ages. Let us discuss lysogeny in detail. The first step in lysogeny is repression. When in lysogeny, the lambda phage enters the host E. coli cell, circularizes its DNA, and its immediate early genes are transcribed and translated by the host RNA polymerase as would be in the case of lytic cycle. But then a 24 kilodalton phage protein, lambda repressor or C1 gene protein appears and binds to the two operators, ultimately shutting down the transcription of all genes except for C1 gene, the gene for lambda repressor itself. Let us now see the details of this particular molecular event. The initial events are common for both lytic and lysogeny. It is later that the two pathways diverge. The host RNA polymerase initiates the transcription of the immediate early genes first. These include CRO, which lies immediately rightward of the right promoter PR, followed by C2, and N, which lies immediately leftward of the left promoter PL, followed by C3, as can be seen in the picture. The operators of these promoters are OR and OL respectively. Initially, no repressor is bound to these operators and hence transcription proceeds without any interruption. When the polymerase reaches the end of the immediate early genes, it comes in contact with the terminators and stops. Let us discuss the establishment of lysogeny. The delayed early genes are not only important for lytic cycle but also for establishing lysogeny as mentioned before. Some of the delayed early genes are required for the integration of the phage DNA into the host DNA, which is the most important step in lysogeny. Secondly, the products of C2 and C3, which are synthesized along with CRO and N, are responsible for the transcription of C1, the lambda repressor, which is the most important component of lysogeny. The C2 protein is a stimulator for the transcription of C1 gene. It binds to the PRE promoter and brings about leftward transcription. This leads to the production of C1 protein, the lambda repressor, which is a 27 kilodalton dimer protein. Lambda repressor, when produced, binds to both the right and the left promoters and shuts off the transcription of all genes required for lytic infection. The only promoter now active is the PRM, that is promoter for repressor maintenance, which keeps producing lambda repressor. It is this lambda repressor which also protects the lysogen from superinfection by other lambda phages. Even if another lambda phage infects this lysogen, the already formed lambda repressor is enough to repress the lytic cycle of the newly infecting lambda phage. Now, the stage is set for the next step in lysogeny, that is the integration of lambda genome into the host genome. Lambda phage DNA is integrated into the host cell genome by site-specific recombination. The site of attachment on the lambda phage DNA is called attP, whereas on E. coli DNA is called attB. The sequence of the site attB consists of the parts B-O, B-, whereas the complementary sequence in the circular phage genome attP consists of the parts P-O-P'. The region O is the 15 base pair central conserved sequence and represents the point of crossing over. The lambda attachment site attP lies between int and J genes, and the bacterial attB lies between the gal and the bio genes, as can be seen in the picture. The attB is only 30 base pair in size and contains the conserved central 15 base pair region where the recombination reaction will take place. Integration of Bacteriophage lambda requires one phage encoded protein int, which is the integrase, and one bacterial protein IHF, which is the integration host factor. Both of these proteins bind to the sites on the P and P' arms of the attP to form a complex in which the central conserved 15 base pair elements of attP and attB are properly aligned, as can be seen in the picture. The integrase enzyme carries out all of the steps of the recombination reaction. The result of the recombination is that the integrated prophage is flanked by the two hybrid attachment sites which are slightly different. attL has the structure BOP' and attR has the structure POB'. The phage DNA is now part of the host's genome. As long as the host cell is doing fine, the virus has no incentive to switch from the lysogenic phase and continues producing the lambda repressor. The phage is replicated each time the host cell undergoes cell division. Thus, the phage gets a free ride. It is the dormant lysogenic state that assures that the phage will not replicate itself into oblivion, eventually destroying all the viable E. coli cells that can serve as suitable hosts. However, if the host cell is exposed to unfavorable conditions such as DNA damage, it produces a stress protein called REC-A. The REC-A cleaves the lambda repressor bound to the right and left promoters. This allows transcription of the PR promoter and an increase in the CRO protein. CRO protein will stimulate the rightward expression of the PR viral genes, leading to the replication and assembly of new viruses, which is characteristic of the lytic phase. In order to induce the lytic cycle, the lambda phage has to excise its genome from the host genome. Excision of Bacteriophage lambda requires two phage encoded proteins, int and cis, which is an excisionase. It also requires two bacterial proteins. In addition to IHF, a protein called FIS is also required. All of these proteins bind to the sites on the P and P' arms of the hybrid attachment sites, forming a complex in which the central conserved 15 base pair elements of the hybrid attachment sites are properly aligned to promote excision of the prophage. The result of this site-specific recombination is excision of the lambda prophage and regeneration of the original attP and attB sites on the phage and bacterial chromosome respectively. One might expect that lysogeny is established every time, but this is not the case as we have just seen. The question is, what decides whether a given cell infected by lambda phage will enter the lytic cycle or lysogeny? There is simultaneous production of CRO protein by the CRO gene and the lambda repressor protein by the C1 gene, both being repressor proteins. Since both proteins can block the transcription of each other, there seems to be a molecular race between the production of CRO protein and lambda repressor protein C1. If C1 wins the molecular race, lysogeny will be established, and if CRO wins, the infection would be lytic. If enough of C1 repressor is produced, it binds to the left and right promoters and operators, preventing the expression of the genes required for lytic infection, and hence lysogeny is established. On the other hand, if CRO concentration is higher, it prevents C1 transcription and hence lysogeny is blocked, making the path clear for lytic infection. Thus, lambda phage can lead two different life cycles. It can lead the lytic cycle wherein upon injection of the DNA into the host cell, the DNA undergoes circularization. The phage genes are expressed, phage DNA is replicated, and many phage particles are produced and are released by lysis. On the other hand, it can follow lysogeny. Its DNA remains integrated into the host genome without any harm for ages. Every time the host DNA replicates, the phage DNA also gets replicated. When the lysogenic bacteria face adverse conditions, the phage excises its genome from the host genome and shifts to the lytic cycle.